Leonard and Ronald Lauder founded the ADDF because of their own mother's experience with Alzheimer's. The genius of what they have done is to have had the vision to bring together the top scientific talent in the world and then partnering with drug companies and the community most affected by Alzheimer's.
My father and uncle correctly identified that there was really not enough being done to address Alzheimer's and related dementias to help bridge the valley between basic research and translating that into actual drugs that are delivered. And the ADDF has helped fill that gap wonderfully.
Our work at the Alzheimer's Drug Discovery Foundation has been critical to the first approvals of disease-modifying drugs for Alzheimer's disease. Our work has really been changing the way diagnosis is made in primary care.
You have to have the ability to find the best people who can make the most out of your investment in them and their efforts. So finding doctors like Dr. Phillip, who has the ability, along with his colleagues, to feel where are these projects going to make a difference.
ADDF recognizes leaders in the field who have embraced the biology of aging as a framework for developing new drugs for Alzheimer's disease. This has helped to accelerate the development of new drugs in innovative ways.
It's always been a dream for me to be part of an organization, a group of scientists that could really write a new chapter in medical history. It's rare. You know, every decade or so, there's such a productive advance in the science for such an important disease.
We're seeing the FDA, for the first time, approve disease-modifying drugs for Alzheimer's disease. It is a new era.
There's something very, very rewarding and fulfilling when hope becomes a reality. I'm proud of my father and my uncle for all they've created and their vision.
My grandmother's legacy also passes on to health and wellness now in the ADDF, and that makes me feel so proud and thankful.
Please welcome the Chief Executive Officer of the Alzheimer's Drug Discovery Foundation, Mark Roithmeyer.
Good evening. Welcome to the ADDF's Inaugural Scientific Summit. How blessed are we, founded by the Lauder family in memory of their beloved Estee. And how blessed are we that 2 years ago, the family Lauder agreed to cover the operating expenses of the ADDF for the next 15 years. So I can say from this stage tonight, every penny you give goes directly to the science that you're going to hear about tonight. Thank you to the Lauder family. Tonight, we have a wonderful scientific summit for you. We have a media star, a major philanthropist, and 9 of the top Alzheimer's scientists in the world. 2 big thank yous before we start. To Heidi McWilliams, our primary chair of this event, whose vision is wonderfully displayed before you. Heidi, you're fabulous. And to co-chair Nancy Goods, who's responsible for the scientific prize that unites all 9 of the scientists you're going to meet tonight. Thank you, Nancy, and on behalf of your beloved Mel. So let's get started. First up, our featured speaker this evening, Sanjay Gupta. He has multiple Emmy and Peabody Awards for his work as Chief Medical Officer at CNN and Special Correspondent at 60 Minutes. He's a 5-time New York Times bestselling author, including his book, Keep Sharp, Building a Better Brain. Dr. Gupta is a member of the National Academy of Medicine, an associate professor of neurosurgery at Emory, and associate chief of neurosurgery at Grady Memorial Hospital. Somehow, with all of that, he still finds the time to be a devoted father to, count them, 3 teenage girls. Ladies and gentlemen, I give you Sanjay Gupta. Thank you.
Good evening, everybody. Thank you for having me. Delighted to be here. I jumped at the opportunity to to talk about this topic. And I think it's a really wildly important topic. And this is an incredible organization doing some incredible work. So I'm really honored to be here. I got to tell you, for the last 30 years of my life, I have been practicing neurosurgery. While many of you may know me as a writer or a reporter, I think of myself as a student of the brain, and I continue to be fascinated and bewildered by the brain. So I jump at the opportunity to ever have conversations like this. There is so much that's going on, so much of what we're going to talk about tonight with regard to what's happening in the brain, from prevention to new treatments, to even what's happening with transhumanism, the idea of using brain-computer interfaces. It's a really, really fascinating time to be discussing all that's happening with the brain. Now, when I give scientific talks, I often start by providing disclosures, any disclosures that I think the audience should know ahead of time. And what I would like to disclose to you tonight before we get started is that probably like many of you, I have been personally touched by Alzheimer's dementia. In my case, it was my grandfather, my mother's father. And I still remember when he was diagnosed. I was a teenager at the time. I was particularly close to him. I'm the oldest grandkid. So as he got older, in particular, I was the person he relied on the most. So we got really, really close. And to be fair, it was challenging to make a diagnosis back in those days. We didn't have the same blood testing or imaging studies that we're going to talk about tonight. And yet when you live with someone and you're close, you're really close, you become pretty certain as well. My grandfather was sort of the natural storyteller in our family. His stories could last for hours. As a child, I would watch the sun literally go across his face from bright sunlight to dark shadows. His stories could take that long. And he spoke this sort of native Hindi and English mixed together, a portmanteau language that he sort of made up, full of words and gestures, and you had to listen with your eyes and your ears at the same time. And he was really masterful at this, so I remember. He would sort of lapse into a whisper every now and then, and you'd lean in close, and then he would clap his hands and laugh at your surprised face. That's who my grandfather was. But then his stories became harder to follow. And again, I know this is going to sound familiar to many people in the room. At first, we thought that he was just telling jokes that we really weren't in on. That's what we thought at first. And then it was clear that his memory started to fail. His expressions started to become more blank, I would say, and he would lapse into these long periods of silence, something we had never really seen before. Most frightening, I think, at the time was the personality changes that came about as well. To this day, I don't know if he was just frustrated with what was happening or it was more a hallmark of the disease, but my sweet, kind-hearted, good-natured grandfather, he started to become more angry and despondent, and at times even violent. And as a teenage kid who was really close to him, it was hard to process. And so I start with this disclosure because part of the reason I wanted to be here today was that this is the reality for millions of people. It's why I devote so much of my reporting to this topic, both in form of books and documentaries. As you all know, neurodegenerative diseases affect everyone. They affect the patient, but they affect the whole family as well. And it can rob people not just of a long life, but can rob them of the essence of who they are during that life. I wrote something in my journal around the time that this was all happening with my grandfather, and it said this: just because my grandfather has become increasingly forgetful does not also mean he shouldn't be remembered. And the people who are forgetful get forgotten. It's just this weird thing. You start to become more forgetful and you become forgotten. That is the nature of neurodegenerative diseases as well. So I really wanted to devote so much of my reporting career to address that issue as well. What happens to people who are diagnosed with the disease beyond the obvious, beyond what's happening in their brains, but what happens to their lives as well. And I think it was a large part of the reason I decided to study the brain. 4 years of medical school. After college, 7 years of training, I did a fellowship, and I think all along the way, I was wondering in part what had happened to my grandfather's brain. How could plaques and tangles potentially lead to all this? Could something more have been done? And I think to some extent, selfishly, you start to wonder, will this also happen to my mom? Might it one day happen to me? I went into neurosurgery because I wanted to fix things. I wanted to remove tumors and clip aneurysms and fuse spines. But there was still this long sense of hopelessness when it came to dementia specifically. The attitude often seemed to be that really nothing you could do about it. In many ways, it seemed like it was treated like a weather event, like a hurricane. You hunker down as best you can, you watch the destruction take place, and you survey the damage afterward. And I think, again, it's going to sound very familiar to many of the people in this room, the belief that those problems then cannot be prevented. And if the damage occurs, it cannot be repaired. But now, thanks to many of the people you're going to hear from tonight, we know that's not the entire story. That's why I wanted to be here today. By the way, I got to tell you, just on a personal note, I left my family's spring break to come join all of you. Hadn't actually told my wife that I was doing this till last night. That's dicey, I'm telling you. I would not recommend that. But when I told her last night that I was doing this and I explained this organization, she even helped me pack my bags. Good wife. I was a little surprised at how quickly she helped pack the bags, I'll tell you. But the truth is, I really wanted to be here to talk about things that I think are happening within the world of Alzheimer's dementia, things that are inspiring and they're exciting. Even little things like this idea that we talk about these plaques and tangles, but we now know that they are present in the brain decades before symptoms develop. So what does that mean? What are we to make of that? The preclinical time, as it is called. Should we think of that as yet another pessimistic, gloomy aspect of the disease? Or should it be a cause for more optimism? That the brain can still function well, even in the face of those growing plaques and tangles. And perhaps an opportunity to intervene early. And we're going to talk about that. Even prevent the clinical symptoms from occurring in the first place. Doesn't necessarily matter what the brain looks like. Matters how it performs. We now know we can produce new neurons at any age. That's a pretty revelatory thing. When I was writing the book and I talked to neuroscientists all over the world, there was this revelatory sort of nature of this. This is not how we were trained. This is not what we believed. Even when I was in medical school, the belief was that you got a certain number of neurons, and that was that. And over the years, you'd slowly drain the cache, and certain things might accelerate that drainage, like alcohol, for example. But we now have plenty of evidence of what this can look like, a very sharp brain at a very advanced age. A few years ago, one evening, I was on call, and there was a 93-year-old gentleman who was brought to the hospital. He was brought to the emergency room, and all that I was initially told was that there had been an accident of some sort. A CT scan revealed that he had a bleed on top of his brain. Now, when my chief resident first told me about this, I won't lie to you, the main fact that stood out to me was his age, 93 years old, and I wondered, what kind of shape was he in? Would he be able to undergo an operation? So I went to the family to have a conversation, fully expecting them to say, look, maybe this advanced operation on the brain, maybe he's not going to be able to tolerate that well given his age. But things took a really interesting turn. I got to the family waiting room, and the spry woman who looked to be in her 60s, I would have guessed probably one of his daughters, I thought, was sort of nervously pacing the room, all the other family members sort of sitting earnestly around her. And I asked if the man's wife was there, and pacing woman came over and raised her hand. Now, I mean, I realized he could have married a much younger woman, but I also knew that they had just celebrated their 70th wedding anniversary. And she looked to be no older than her 60s. Sort of sensing my confusion, she said, I'm actually older than he is. I robbed the cradle. She was 94 years old, in perfect health, took no medicines, was still driving. In fact, had driven her great-grandkids to school earlier that morning. Her husband, my patient, the 93-year-old, I was told was even healthier than her. He was still an avid runner, and he still worked part-time as an accountant. They kept him around, his 65-year-old son told me, because he was such a whiz with numbers at 93. His brain bleed had occurred, in fact, because he had taken his leaf blower to the top of his house and was trying to blow the leaves off of his roof. These nonagenarians, as they're called, people in their 90s, were healthier than most of my patients at any age. So what to do? So given all that, given that he was physically and cognitively healthy and that was living independently and the family wanted to proceed. I took him to the operating room for something known as a craniotomy, brain operation. You make burr holes in the skull, you take out a flap of bone, and you're then looking at the dura, which is the outer layer of the brain. You open that up, and there was a blood collection there. It was more viscous than liquid, so it was causing pressure on the brain. Remove that blood collection, stop any remaining bleeding, and the critical part of the operation is done. His brain is decompressed. All that's left to do is close the dura, put the bone back, and suture the skin. But before I did that, I decided to take a couple minutes to inspect the brain, just take a close look at the brain. And what I saw was really interesting. Given how sharp he had been, how active and cognitively intact, I expected to see this large brain pulsating robustly, looking really healthy. And what did I see? I saw a 93-year-old brain. It was more shrunken. It had these deep crevasses in it, indicative of his age. Now, maybe you hear that and it sounds disheartening to you, but this is my entire point. It should feel just the opposite. Doesn't matter what the brain looks like, matters how it performs. Yes, of course his brain had aged. He was 93 years old. But the brain, perhaps more than any other organ in the body, can reliably stay sharp throughout your entire life. I say that out loud, but it's an extraordinary statement, one that we really didn't think about for a long time. I'll never forget there seemed to be this total disconnect between the brain I was staring at in the operating room and the man whose skull it inhabited. As you might imagine, I was anxious to see how he would do from the operation. I went to go see him afterward. He was in the ICU. There's no TVs in the ICU, but he was sitting up in bed looking at his smartphone. He had his reading glasses sort of adjusted over his head bandage, and I was curious, had I extended his life or extended his death? When I asked him what he was looking at on his smartphone, he said he was following the elections in West Africa, as one does right after emergency brain surgery, apparently. But it turns out he had been there just 10 years earlier doing volunteer work when he was 83 years old. He was recovering really well. And after I examined him, I asked him, sat down on the side of the bed, and I asked him, wondering how the whole event had sort of affected him, how he thought about mortality overall. He smiled and he looked at me and he said, I think the biggest lesson in all this, no more trying to blow the leaves off the roof. The guy taught me a lot. We think of him as the exception to the rule. But in many places around the world, he is the rule. He's the rule. He lives the life that people who you're going to hear from tonight prescribe. And it leads to an extraordinary sharp brain, even at an advanced age. A life full of movement, brisk movement, perhaps more than intense movement. It's your best chance of releasing something known as brain-derived neurotrophic factor, BDNF. For people who follow this, that's the miracle grow some people call in the brain. A healthy diet, particularly low in sugar. If you're going to do one thing, just start to eliminate excess sugar. Just do that. That'll get you a long way towards getting to a better diet. Prioritizing sleep. Best advice, leave your phone in another room. And make sure the first light that hits your face in the morning is natural light. So open the drapes before you turn on your phone. And connect. Self-described loneliness was associated with a doubling of the risk of Alzheimer's. One of the easiest ways to connect, by the way, sort of a hack, is to allow yourself to be vulnerable around other people. Something loneliness researchers will tell you, it's sort of a hack. People always say it's not the quantity of friends, but the quality of friends. How do you measure the quality of a friendship? Are you able to be vulnerable around that person? Whenever I come out of the operating room, I see a waiting room full of people, I'm instantly more optimistic about my patient's recovery. I spent a long time working on Keep Sharp. I traveled the world, talked to scientists all over the world. This took place over years, and when it was finally done, I called my mom to tell her I'd finished writing the book. And my mom said, can you summarize it in a couple sentences for me? Moms. I told her that as much as possible, she should try and take a brisk walk with a close friend and talk about her problems. It's interesting, the prescriptions that we have for a healthy brain, very different than what you'd often expect. I spent a lot of time looking through our collective medical history when working on this project. And there was a study, the Framingham study, which you'll hear a little bit about tonight. But the Framingham study on heart disease about 75 years ago kind of changed the way that we think about heart disease. We used to think of heart disease much in the same way that we think about brain diseases today. Like that hurricane that comes and there's nothing you can do. Now, most people, I think, are pretty comfortable with the idea that we can predict heart disease, that we can treat heart disease, and yes, that we can even prevent heart disease. Well, the evidence is accumulating that we're going to be able to say the same about Alzheimer's dementia. It's exciting. These are nascent days, but with both heart disease and neurodegenerative diseases, we know that you can decline very quickly. But we also know that you can improve very quickly. It has taught us something that I think is critically important, which is that we human beings are incredibly biodynamic. We can change very quickly with pretty simple lifestyle changes. One of my mentors once told me that as physicians, we must always promise not to overpromise. And I think I would emphasize that today. Keep that in the back of your mind as you're listening to all these presentations. And yet know this, I am excited, I am optimistic, and I am truly honored to be with all of you today. So thanks for having me.
Thank you.
Thank you. Yep.
Thank you, Sanjay, for sharing your personal stories as well as your unique professional perspectives. Now it's time to pivot to our Goods Prize winners. Last year, the ADDF gathered in Stockholm, Sweden at City Hall with the Queen presiding over the 10th anniversary. She stated then that the recognition of scientific excellence is important, as is the stage you celebrate it on. That evening in the Golden Hall, the U.S. ambassador to Sweden declared the Goods Prize has now become the Nobel Prize of Alzheimer's. It thrills me to let you know that in a few moments, you will meet these Nobel or Alzheimer's laureates. But first, I'd like to introduce you to the moderator of such a prestigious panel. His name is Niranjan Bose, but like the rock star Bono, he goes by one name, Bose. He's the managing director at Gates Ventures. He is the lead on Alzheimer's and is Bill Gates's right-hand man. And in fact, Bose and Bill have raised with the ADDF collectively over $100 million for something called the Diagnostic Accelerator that you'll hear more about tonight. Without further ado, Bose.
For our first segment, Drug Development, please welcome our scientists. From the University of Nevada, Las Vegas, Dr. Jeffrey Cummings. From the University of Arizona, Dr. Roberta Diaz-Brinton. From the Stanford University School of Medicine and Pharmacotropics, Dr. Frank Longo. From Northwestern University, Dr. Marty Watterson. From Vanderbilt University, Dr. Jerry Rook. From the Washington University School of Medicine, Dr. Miranda Orr. Co-founder and Chief Science Officer of the Alzheimer's Drug Discovery Foundation, Dr. Howard Fillett.
All right. Good evening, West Palm Beach. My name is Sanjay Gupta. All right. All Indians do look alike. My name is Niranjan Bose. It's an honor and a privilege to be with all of you. I'm thrilled to be hosting this panel with this amazing group of rock stars that we have assembled here. Before that, let me thank the Lauda family Heidi Tom McWilliams, thank you for pulling this together. And Nancy, thank you for the Goods Prize. Thanks to your family. I'm also honored today, and my parents will be extremely proud of me because I'm going to call them in India and tell them Sanjay Gupta was the opening act for me. Thank you, Sanjay. All right. All jokes apart, the journey with ADDF for us began about 10 years ago. Bill Gates was interested in using some of his philanthropic capital to advance some of the bottlenecks that the field was facing. And we looked around to find a place to move the needle on biomarkers and blood-based diagnostics. We chose ADDF, and that's how the Diagnostics Accelerator was born. We chose that for a variety of reasons. The two key reasons were their scientific rigor and risk-taking approaches to innovation. If we were to do this again, we would do it 10 out of 10 times. We ended up raising $100 million. Approximately $90 million of that has been invested in a portfolio of companies, and we are this close to a blood test. This close. There's still a lot more to be done. You'll hear a lot of the progress being done by these leading scientists. And I got to know a lot of the Goods Prize winners through interactions in Stockholm and before. And I'm more than optimistic that we are going to find a cure for this disease. With that, let me turn to my rock star panelists here. Welcome to my living room. Make yourself comfortable, because there's going to be some uncomfortable moments here. Growing up in India, I was trained to be an infectious disease doc. When I was asked to help lead the Alzheimer's program, these are the scientists whose papers I read, trying to come up to speed on where we should be putting our investments, who should we be working with. And now I have the great privilege of quizzing them, asking them the questions that they would have asked me if I was a PhD student or a postdoc trying to defend. So I'm living the dream, and this is to all the PhD students and postdocs out there. So I asked my friend ChatGPT to come up with one word to describe each one of them. And this is what it gave me. Howard Phillip, innovative. Jeff Cummings, rock star. Frank Longo, game changer. Robbie Brinton Diaz, pioneering. Marty Warrison, trailblazer. Miranda Orr, groundbreaker. Jerry Rook, trendsetter. The other two panelists will join us shortly. Rhoda O, visionary, and Mia Kivipelto, prevention champion. So this is the band of rock stars that I'm going to be playing with tonight. Thank you for joining me in my living room. So before we get started, I'm going to ask you two rapid-fire questions. And the first one is, let's get to know you a little better, your personality. And I'll go around and ask each one of you to give me a quick answer. Are you a glass half empty person or a glass half full person? Miranda, let's start with you.
Half full.
Howard.
I'm glass half empty, but I need a bigger glass.
Good one. Jeff?
I'm half full for sure.
Bobby?
Half full.
Frank?
Half full for sure.
Barry?
Half empty.
Half full for sure.
Half full. Well, it's good, because I'm mostly the half-empty person in most of these audiences. It's good I'm joined by some of my colleagues. The reason why I ask is, put that in perspective as you hear their answers, because that is what we want, a balanced perspective on all of the progress and where we need to go. The next rapid-fire question for you is, if you were not a rock star sitting here, i.e. you didn't go down the path that you are today, where would we find you, or what would you be? Let me start with you, Marty.
I really have no idea because I've wanted to be a scientist since I was a very young age. So this is the ideal profession for me.
All right. Jerry?
I wanted to be a neurosurgeon when I was a little kid.
Frank?
Well, I spend quite a bit of time doing research now. I think I would be a full-time clinician. I love medicine.
Robbie?
I would be a neuroscientist, which is exactly what I am. And once I discovered neuroscience, the game was over. It was like, I'm choosing you. This is what you got to do.
All right, Jeff, you got to give me something other than neuroscience.
Well, I'm breaking the mold here because I'd be an architect. I love to build. I love to see how things fit together. I love to create habitations for people where they can thrive. And that's what I do in science. And that's what I would do as an architect. So I would love to be an architect.
Howard?
Yeah, I've been a physician scientist for over 45 years. I've been very, very fortunate to have a field which I really love my work, and I think that's a great thing to be able to. So I'm not sure I would want to do anything else at this point, but I've been very privileged to be in the position that I've been in these 25 years with the foundation.
Great. Miranda?
I love being a scientist, but if I would have not had this path, I would have been a fourth-generation farmer and rancher in northeastern Montana.
Oh, wonderful. All right. And I'm going to give you some homework that I'm going to check at the end of the panel. Imagine you're writing a note to your family that's going to be opened 25 years from now. And the opening line is, I worked in Alzheimer's and related dementias, and in 25 years, fill in the blank. You don't have to answer it now. You have about 45 minutes to think about it. I'll come in the closing to hear all of your notes. So the first panel is on drug development. The ADDF works on drug development, biomarkers, and prevention. And we'll start with the drug development panel. They don't need any introduction. I'll just give you some context. You've all read it in the news. We have some game-changing disease-modifying drugs that have been approved in the last 12 to 18 months. Game-changing disease-modifying drugs. Huge progress for the field. The ADDF has played a catalytic role in getting some of these, and we're joined by colleagues from Lilly here who've actually played an even bigger role in getting the product to the market. Let me come to you, Jeff. And you've seen this space evolve. You track this very closely. What does the approval of Kisunla and Leqembi mean to the field? And if you are talking to a family, a caregiver, a patient, what do you tell them about the approved medicines?
It's such an important question. And let me just amplify one point that Bose was making, which is it was between 2004 and 2021 when we had no new drugs for Alzheimer's disease. There was a 17-year hiatus during which there were no new therapies. So it's so important that we have these new drugs, and so terrific, but they are difficult to present to the patient. And ultimately, this is about the patient. It's about how can we help the patient, and we want to make sure that they understand what these drugs can and cannot do. And I think the best way to communicate this is how long can they stay in a highly functional state. So we would say that if a person is on one of these new drugs for 18 months, they will stay at a higher functioning level for 5 additional months. So it means that the delay in the loss of function, the loss of being able to control their own lives has been given back to them. for another 5 months. And the beauty is that we believe that the longer people are on these drugs, the greater that gain turns out to be. So it's about giving life back to an individual who is on the track of Alzheimer's disease dementia. And that's the discussion that I would have with them. Of course, there would be discussions about safety and monitoring, but when a patient comes to you, they want to know, why would I do this? What would I get from it? And that would be my answer, Bose.
Thank you, Jeff. This degree of optimism that these drugs provide is exactly what we were looking for as a field. Of course, some of them would say it's a modest benefit, but it's okay. They can be naysayers. Howard, you've been a strong champion advocate for us to embrace biology of aging. Given the shot in the arm, where do you see us going in terms of combination therapies and beyond?
So it's very clear and well accepted that aging is by far the leading risk factor for Alzheimer's disease. And aging research has been going on for well over 100 years. The Imperial Cancer Trust, which was one of the first organizations to foster cancer research, was established in London in 1905. So there's been about 120 years of cancer research. And today, after all those years of research, we think of most cancers as a chronic disease that is manageable. Alzheimer's disease wasn't recognized as an illness in older people until 1968, even though it was described by Alzheimer in a 52-year-old woman who was clearly demented in 1906. Nothing was done because it was thought that Alzheimer's was a disease called a pre-senile dementia. When I went to medical school, I never heard the word Alzheimer's disease. It wasn't in our textbooks. We never used it in the clinic. We used to just call people senile. And senility was accepted as a normal part of aging. So there was no research. When my mentor started the National Institute on Aging, which today is the major funder of Alzheimer's research with a budget of about $4 billion a year, when my mentor established the National Institute on Aging as the primary funder of Alzheimer's research in the world, we were spending billions of dollars on cancer research, and we spent $620,000 on Alzheimer's research in 1976. So we've been a late starter, but if you think about how long it's taken us to go from when the beta amyloid peptide was discovered by Glenner and Wang in 1984 to today, it's about 30 years, and it's been established as a metric that it takes about 30 years, maybe 35 years, to go from a discovery of a new target for treatment to the time when drugs become available on the market. So if you take the Alzheimer's research field in context, it's clear that we're right on time today because it's been about 35 years since beta amyloid was discovered, about 30 years, 40, almost 40. The reason I say that is because we have, for the first time in human history, treatments for Alzheimer's disease that are based on the pathology of the disease, but not on the biology of the disease, not what necessarily might create the plaques, but the fact that we just want to get rid of them and see what happens. And it's worked, there's no doubt. And it's amazing that for the first time in human history, we have drugs that can treat senility. But we know that there's much more that's needed because we're going to need combination therapy. And we were going to learn that from what are those new targets? It's going to be from the biology of aging, everything that's been learned. For example, inflammation is recognized as one of the hallmarks of aging. And it's finally being recognized as the leading target for drug research because we see even Alzheimer described inflammatory cells around the plaques. But for the first time, we have this accepted as a reliable drug target. And I think the next phase of this is going to be learning from aging research on why inflammation goes out of control, developing drugs to manage inflammation, and it's tricky, in the aged Alzheimer patient. Metabolism, insulin resistance, GLP-1 agonists, for example, and about 8 categories, including senescent cells. It's interesting, I'll just close by saying that the goods prize representatives that you see here represent the 8 or so major pathways of aging in the biology of aging. So our portfolio among the goods prize winners really does reflect clinical and basic preclinical research on the biology of aging, and we hope to build out that portfolio of drug discovery based on getting these two fields together, getting neuroscientists together with what we call biological gerontologists, scientists who study the biology of aging, translating all that research that's been done well over 100 years into new drugs for Alzheimer's disease.
Thank you, Howard. Now that we've touched on the game changers, the disease-modifying drugs, I want to come to each one of you. What are you most excited about that you're seeing in the field of clinical trials in this space? And a second part question, what's going to be the next game changer for the field? Start with you, Miranda.
What I'm most excited about is what Howard was just talking about, which is the biology of aging and combination therapies and repurposing different therapeutics for the treatment of Alzheimer's disease. So in the biology of aging, a lot of medications that many of you may take or may end up taking at some point in your life, like metformin, these medicines extend healthy lifespan. And we've known this in the biology of aging field for many years. And so now really understanding it and utilizing it in the context of Alzheimer's disease is incredibly powerful, and it shortens that amount of time between identifying a target and getting it into people to help them. In our particular program, we are studying cellular senescence, and I know that's a big word, but this is a type of aging cell. It's one of the hallmarks of aging, and these cells are major contributors of inflammation. And we found a link between Alzheimer's disease and this hallmark of aging, and the ADDF is funding our trial for the first time to test a therapy to clear these senescent cells in older adults with Alzheimer's disease and look for benefits. And so without the ADDF and really supporting innovative research in early stages, we wouldn't be running this trial.
Thank you. Marty, let me come to you.
Well, I think the inflammation part, in a way, it's an enlightened self-interest of mine, but it is now actually accepted as mainstream. A few years ago, this was considered interesting, but a bit naive about the disease progression. At this point, I think ADDF has made the intellectual contribution to the field towards that, but also, for me personally, it's been the catalytic funding where they gave us seed money to be able to develop novel small molecules that were multiple shots on goal for the inflammation, what's called a synapse, which is the basis of cognition and behavior, that interface. And I think that interface, that systems biology, is going to be exploding because of some of my colleagues in the Goods Prize who develop biomarkers. We have biomarkers for the disease, but we now have biomarkers for the inflammation process, and we can now monitor those as we develop these novel therapeutics to show that we're engaging them. And that's going to be another major area of growth, I think, in the coming couple of years.
Thank you, Marty. Jerry.
I think
and don't forget the game changer, please.
I'm sorry?
Don't forget the second game changer.
The game changer. I think one of the things that I'm most excited about with clinical trials is we've made some significant milestones in the last decade or so, and one of the huge hurdles we've overcome is being able to select patients that should participate in these trials. So before, we didn't really have the ability to predict who was going to develop Alzheimer's disease or progress further in the disease, and that really hampered our ability to test these disease-modifying new compounds that we were, trying to develop into drugs. And so I feel like now that we've made such significant progress, we've got brain scans, blood-based biomarkers that really increase our confidence of who may develop Alzheimer's disease. And so now we're really poised at this point in time to test all of these theories, all of these potential cures or disease-modifying medications that we're trying to develop. Me personally, I focus a lot on neuroinflammation that Howard and Miranda both talked about, and Marty. But there are just so many different avenues, and it is a multifaceted disease. There's not going to be a magic bullet or one pill, and Howard alluded to this as well. It's gonna require combination therapy, personalized medicine for each individual, and I feel like at this point in time, I feel we're going to see a significant increase in the success rate of our clinical trials.
Thank you. I know Frank's getting comfortable. I'm going to go to Jeff.
Okay, two things stand out to me, Bose. One is we have success in monoclonal antibodies. That's a drug that we were talking about just a moment ago. And we're not at the end of that era yet. There are changes going on every day to make those drugs better, easier, safer. So there's a whole story to be played out that we're already have in process. Fantastic. And then I just want to play this inflammation theme that we're developing here a little bit more, because Ozempic, which you've all heard about because it's treatment for diabetes and the treatment for obesity, is a profoundly anti-inflammatory drug. And we think that the reason it's helping in diabetes, it's helping in obesity, it's helping in chronic kidney disease and in cardiac disease is because of the effect of inflammation, and that might translate into a therapy for Alzheimer's disease. And that trial will read out this year. So we're going to know by the end of 2025 whether Ozempic is also a treatment for Alzheimer's disease.
Fantastic. I'll go to Frank.
Yeah, in terms of trials, one of the areas I'm most excited about is just the diversity of targets and mechanisms we're going after now. So in recent years, we've learned much more about Alzheimer's and how the synaptic connections in the brain are lost. That's why we lose our memories and our cognition. So since we know so much more about those mechanisms now, many of us are developing treatments that can go after those mechanisms to prevent the loss of these connections. And we're starting to see those enter the trial space now. I'm very excited and optimistic about that. And then what I think that will lead to, and this will be a game changer, I know our own program is working on this, we're understanding resilience more and more. And Sanjay Gupta brought up this really critical concept of brain resilience. Just last week, another patient was reported in one of these hereditary Alzheimer families that have the most severe forms of Alzheimer's. They're demented by age 40. The brains are full of amyloid, and in this case, yet another report, but the patient had normal cognition. The amyloid was not capable of causing this loss of our synaptic connections. We're developing a drug that we think can confer that resistance, and so that allows us to see the light at the end of the tunnel. We can confer resilience on people. They don't have to be just lucky, and maybe we can apply that well before the symptoms start. That'll be the game changer.
Fantastic. Robbie, bring us home.
All right. Well, I think a number of folks have mentioned that there's this convergence now of advances that have been in the making for 20 to 30 years in brain imaging, in blood-based biomarkers, and in therapeutics. So one of the things that I think ADDF has been an absolute leader in is precision medicine for Alzheimer's disease. And that requires a portfolio of therapeutics. And ADDF has supported development of that portfolio that it will enable, is enabling and will enable precision medicine for Alzheimer's disease. And our therapeutic, which was supported in the very early stages and continues to be supported by the ADDF, is coming back to something that Dr. Gupta mentioned, is neural stem cells. And we're developing the first therapeutic to regenerate the Alzheimer's brain. And we're very thrilled that in our early stage clinical trial, we had encouraging evidence for regeneration. And we're now in a phase 2 clinical trial. And we're very excited and very grateful for the support over a very long period of time and for the innovation and the support of innovators through the ADDF.
Thank you, Robbie. This is some exciting times. We're all in. A lot of this progress in therapeutics, treatments, the advances in clinical trials wouldn't be possible without biomarkers. Let's switch gears, shift our segment to the biomarker segment, and let's welcome Dr. Rhoda O. Rhoda, welcome to my living room. My friends here would like to get to know you a little bit better. Are you a glass half full or a half empty person?
I'm definitely half full. I actually would say I'm 3/4 full.
And if you weren't doing what you're doing today, where would we find you? What might you be doing?
I actually think I would be, albeit a burned out one, a management consultant. And the primary reason, I like to solve problems. I actually really like the concept of giving a problem and then trying to figure out what the solution is. And I think that's very consistent with being a researcher.
I used to be a management consultant, and I quit that job because it was very boring. I was doing the same thing over and over and over again for different pharmaceutical companies.
All right.
I'm glad they started the clock because I was afraid I was going to be out of a job soon because that clock was not working. And before I came on stage, somebody sent me a text message, one spammer, there were part-time jobs available. Thanks for starting the clock. Howard, blood-based diagnostics and tests, they're a near-term reality. How does it change the field?
I think a few years ago, if you had asked many of us if there would be a blood test for Alzheimer's, we would say that's impossible. But today, blood tests for Alzheimer's disease are on the market in 49 states. The only state that's not approving them yet, and it's thought to be approved soon, is New York State. But they're already out there. They're excellent. They're well validated. They have good accuracy. And I think they're going to really revolutionize clinical care. Imagine if we tried to develop drugs for heart disease and we didn't have a cholesterol, or drugs for hypertension if we didn't have a blood pressure cuff. Those are examples of, really simple examples of how biomarkers play a critical role in diagnosing disease and treating the disease and developing drugs to treat the disease. And now we're at that point where with the blood tests that we have on the market, it's going to change clinical care, it's going to change prevention, and it's going to change the way we enroll patients, because the current way that we did it before was we didn't have any way to be certain to identify people that had Alzheimer's disease. So it was really a clinical judgment. And it was shown that about 30% of the time, even experts in dementia got the diagnosis wrong. So these blood tests are really going to revolutionize clinical research and care. And the ADDF, the diagnostic accelerator, is on the verge of looking at a worldwide validation and quality control mechanism to make sure that whether you get a blood test in France or on the West Coast of the United States or whatever it is, that there's quality control, that results can be compared. And a patient comes into my office today and they're 60 years old and they want to know if they're at risk. I can do a blood test. It correlates very well with the brain scan. And I can give them some diagnostic information rather than not really being able to tell them anything at all. Because we know from these biomarkers that we have, the brain scans, the blood tests, that it takes about 20 years between the time that somebody starts developing the pathology of the disease and the onset of symptoms. And so the blood tests and the brain scans have enabled really rigorous precision prevention as well. And I think it's really a revolution.
So what does the blood test tell me? Go get a scan, or does better than that?
Right. We're really not there yet in terms of saying whether the blood test is enough. I think we're not quite there yet. I think there are good correlations and studies being done now. What is the role of the blood test in primary care? We don't really know yet. And when should the blood test be approved or proven to be correct by, let's say, a brain scan? I don't think we have all of that worked out yet because these tests are just coming to market.
You're definitely a glass half empty person. I'm going to disagree with you and say, I think the blood tests are here.
They're here, and you can go to Quest, you can go to LabCorp and get the blood tests today in your state. I practice in New York City, and I can't get the blood test. That's very frustrating, but it'll happen. I know it's going to happen, and I think it's really going to change the world.
2024, 2025 could be said as the year of the blood test for Alzheimer's disease. Rhoda, let me come to you. In the coming years, what do you think will be the advances in biomarker development broadly in the field?
So I think we actually need to get back to the clinical expression of the disease. So it's one thing to have the biomarker, but it turns out that if you have pathology, you won't necessarily express it. And so I think what's really critical is we've got to do better now in detecting it from a clinical standpoint. So I think the future in biomarkers is really this idea of digital biomarkers. And I think that I'm actually really optimistic about the potential of digital biomarkers, even though there might be still some skeptics about it, just because we didn't imagine blood-based biomarkers 10, 15 years ago, and here we are. I think the big difference with digital is I think we can get there faster. So I like to think that we're going to get there in the next 5 or so years.
Just comment.
Please go ahead, Howard.
Well, the digital biomarkers give us clinical information, and that's essential to making the diagnosis. It's like when people ask me, well, what's the difference between dementia and Alzheimer's disease?
Sure.
So dementia is a word that we use to describe the patient. They have to have cognitive impairment that's progressive and chronic and isn't caused by drugs or depression or something reversible. But it describes the patient. And I agree with you that we need to be able to evaluate patients on their cognition in a more rigorous way using digital biomarkers. But the blood tests don't tell us about the clinical state, they tell us about the cause.
Sure.
The potential cause. So the first thing I would do is try to figure out in the first place what the person's cognition is. And then if it was impaired, I'd do a workup to determine what the cause is. And now I can just do a simple blood test and see if the probable cause is Alzheimer's disease. And what's even better than that is people used to say, why bother doing the blood test if there's nothing you can do about it? But now we have 2 disease-modifying drugs on the market that actually probably are going to require the blood test in order to get the drug. And so it's just the beginning of a new era.
Yeah.
I'm going to go to the defendant.
Yes, because I was going to say, so I think one of the key things that you said is that you're going to wait until they have that clinical symptom, and that's what I'm very interested in. And so you think, it's sort of this idea, is the biomarker here of the disease and the clinical onset 20 years from now? I don't think it's going to be 20 years from now. So one of the things I'm very interested in is digital voice, right? Because I think that speaking is a very cognitively complex task. We all speak, and I think Dr. Gupta talked about his grandfather and his being a storyteller, and how it was the change in the storytelling. That's digital voice, right? Speaking, that they started to pick up that signal. And I think we're going to be able to do that on the digital front. So we'll be able to actually trigger the blood test to follow because we're picking it up on the behavioral side much earlier.
There's so much digital data out there, smartphones, watches, Fitbits, rings, etc. How soon can we get a multimodal test that could either predict accurately or give me a risk score?
So I think that, so again, I was the 3/4 full. So I actually think that we're not going to have to wait as long as we did with blood biomarkers. I do think we can shorten the time. There are 38.6 billion internet-connected devices right now worldwide, and those are all digital data streams. So our challenge now is how do we capture all this information that we're now gathering and put it through now these advanced analytics that we have with the data science AI to get to that reality. So I think that the speed in which we can collect this data and do it on a worldwide basis will allow us to get to that reality of digital biomarkers faster than the pace that we got to blood.
You mentioned digital voice. I believe it could be a powerful signal, but there's a lot of concern about privacy. That you can re-identify somebody by their voice. Your thoughts on that?
Yeah, I think that, well, anything in digital, we have to worry about sort of the privacy, confidentiality. So one of the things that we're doing, and we're actually doing this through the Global Research and Imaging Platform, is we're developing these tools in order that we can de-identify these digital voice so that we can more broadly be able to use them while protecting people's privacy. So we do have to do some work around sort of de-identifying, privatizing these data. But I think we'll get there, and I think we'll get there within the next year or two, at least at some level, and then that will break this open in a way that I think is going to be exponential in the speed in which we get to discovery.
Fantastic. On that note, I'm going to keep us moving and move to the next segment. But Rhoda, did you have a chance to hear the homework?
The homework, yes. You said something about 25 years from now?
You're writing a letter to your family 25 years from now. It's going to be opened.
Yes.
And it opens with saying, I worked in Alzheimer's.
Yep.
And ... You fill it up. You don't have to answer it now. We'll close it with
All right, I got it.
everybody's imaginary letters opened.
Okay.
Prevention. This is the third and last segment of our panel. We will have treatments. We heard about the advances in biomarkers and diagnostics. Prevention is very important as well. Secondary prevention, ultimately primary prevention. And one of the Goods Prize winners, it's a great honor to welcome Mia Kivipalto, who has been a champion of prevention. Mia, while you get comfortable in the living room, 2 quick questions for you. Are you a glass half empty person or a half full person?
Absolutely half full person.
And if you were not the rock star that you are today, that the world looks up to, what would you be?
You know, I can't think anything else what I'm doing now. I have always wanted to be a researcher, study brain. I'm a medical doctor, and if I would not be a researcher, I would love to work with my patients, and I do it as well. So I have been able to combine both the research and work as a medical doctor.
Well, you're also committed and dedicated. I wish I could have been a cricket player instead of what I'm doing now. Mia, you led one of the pioneering studies in the world, the finger study. Share a little bit more about why did you embark on the finger study? Why was it so important, and why the findings are very critical to all of us?
Yeah, thank you, Bose. And indeed, I would say that prevention field has been advancing so much during the last years. When I started my studies more than 25 years ago, it was only high age and genetics. They were the established risk factors. Now, thanks to the research supported by ADDF, we know that at least 45% of all dementias are linked to lifestyle, health, environmental risk factors. So there is a great opportunity for reducing the risk. And we also know, as we heard, that the process leading to dementia may start to develop 20 years before we have the symptoms. And that's why we normally say it's never too early to start to prevent. It's easier to prevent than repair the brain damage. So the finger study was the first proof of concept that it is possible to prevent cognitive decline with lifestyle-based interventions. And what we did, we developed and tested the concept multi-domain interventions. It's not enough with one single intervention. You need to target several risk factors and mechanisms at the same time. So finger name, it reminds us about the interventions, what we had in the finger study. Healthy diet is the first finger, very important for the brain throughout the whole life course. Second finger is the physical activity, and that's one of the most powerful interventions. We had aerobic exercise, muscle training, and balance training. And you know, already 10 minutes of exercise can release almost 10,000 molecules in the body. So that really affects the brain. Third finger is the cognitive training, and we heard about the cognitive reserve. What we are doing is affecting the brain, and we can really increase the resilience in the brain. Fourth finger, that's the social activity. That's so important for the brain health and well-being. And now we have been adding stress management and sleep to that fourth finger. And the final finger, that's taking care of all vascular metabolic risk factors, diabetes, we heard about that, inflammation, blood pressure, lipids. That can be both non-pharmacological and pharmacological interventions. So when we did that for 2 years, we could see clear effect on memory, executive function, total brain health. And also the good news was, I often get the question, how about if I have the genetics, the genetic risk profile? So we have been able to show that if you have APOE4, the risk gene, the benefit of the finger can be even higher. So I think there are many, many good news what we can do already now. We published the finger 2015 in Lancet, and 2017 we launched the Worldwide Fingers Network. And I'm happy to tell that now we are having 70 countries who are part of the network, developing, optimizing the model. And thanks for the support from ADDF, we are upgrading Finger to Finger 2.0, that we can combine lifestyle interventions with pharmacological interventions. So I am optimistic for the future.
That's wonderful, and thank you for sharing the lifestyle intervention story there. But we all know behavior change is hard. Lifestyle interventions are very hard for all of us, including me. Howard, when can I pop a pill? What about drug?
Well, again, like heart disease, if somebody is diagnosed with coronary artery disease, they're given healthy lifestyle, maintain interactions socially, and do all the things that Mia described for the brain, plus they're given a statin. And I think that's the way we're going to prevent and treat Alzheimer's disease, is lifestyle interventions, management of the comorbidities like diabetes and hypertension, and taking the pill. And through precision medicine, we'll do a biomarker profile of the patient, see what mechanisms are involved, like inflammation, maybe yes, maybe no, and then giving the people the pill plus the anti-amyloid to remove the plaques and get rid of the pathology that might be contributing to the inflammation in the brain.
That's great. Before I come to you, Mia, with the same question, how many of you would prefer to have a pill? It's okay, pharma company, you can also put your hand up. Mia.
Well, I think it's the combination, and that's what I really believe. Starting always with lifestyle, with small steps, and that's the secret, I think, with the finger model, that you don't always need to do huge changes. Increasing the positive things and decreasing the negative, you can really have a huge impact. And that's what we are now studying in the MedFinger trial, supported by ADDF. We start with lifestyle, and then we add medication when needed. So the precision prevention, the right person gets the right treatment at the right time. We started with metformin because it's widely available drug, and the combination or the links between diabetes and dementia. Now we would like to start and test new treatments. GLP-1 was mentioned. So why not to use this as a platform design, adding new arms depending on the risk profile? I really think the concept precision medicine is the future. One size does not fit all. We need to better find the right person and the right target.
Precision medicine is absolutely critical here, given the heterogeneity of the disease. Some people say, my Alzheimer's is not the same as your Alzheimer's. Howard, as we think about prevention, where do you think the field will be in 5 years or 7, 8 years?
Well, I think, as you mentioned, Bose, compliance is a key issue. And I think that was part of the value of the finger study, is that it encouraged compliance. One of our board members, when we told them that exercise can actually improve cognition and other biomarkers that we measured in the clinical trial we funded, the person on the board said, well, that's fine, but I'm a couch potato. Can you please give me a pill someday? But I think the next phase of this is implementation. And what we're starting to see, this is happening in Europe and other parts of the world, as well as in the United States, is the development of brain health clinics in your mall, on your street, in the downtown area, where compliance will be part of the prevention and compliance of brain health will be part of everyday life.
That's great. We have a couple of minutes before I need to come to closing. So I'm going to come to all of you. It's a 15-second answer at most. because you're going to be riding an elevator with some of these donors. You've got 15 seconds before they reach the floor. What should they be contributing towards? 15 seconds, please. Who wants to go first? All right, Frank.
The ability to accurately measure in a living person the status of their synapses.
That's awesome. All right, Rhoda.
Digital is the new blood. And when we collect digital, we are going to enable, just like we do with blood, many different markers of disease.
Jeff?
I would say a blood test that allow precision medicine, so they tell us enough that we know what drug to use.
Yeah.
We need clinical trials to really show that something works. And I would like to develop the next generation of clinical trials, the platform, so that we can speed up the development. As I testing all these different pharmacological interventions at the same time. We can't wait 20 more years. We should do it in 2 years or maybe 5 years.
Wonderful. Marty?
Well, we have, as people have talked about, things on the shelf ready to go now that are precision medicine oriented, that are better next generation versions of these effective clinical drugs which have issues. So I agree, clinical testing of those hypotheses using biomarkers and these new precision medicine oriented candidates. And within the next 2 to 5 years, I think this will be a revolution of where we're going.
That's great. Miranda?
I think investing in measuring biological age, not chronological age. We need to know the age and the biology of a person.
Joey?
I would like us to be able to reduce systemic inflammation throughout the whole body. I think it's as important for brain health as it is for all the rest of our bodies. So,
Robbie, I'm going to come to you next.
We will be able to regenerate that Alzheimer's brain and regenerate that life.
I'm going to come to my friend Howard and remind him it's 15 seconds.
Well, I'm going to take a heretical position. And if I was in the elevator for 15 seconds, I'd tell the person, ADDF is about all of the above. Invest with us. We're the only venture philanthropy out there for Alzheimer's disease. We've funded over 750 drug programs in 20 countries, including 150 biotech programs. And I think if you want to make progress, we have a staff of 15 neuroscientists on staff that are every day doing due diligence. And
the elevator door is opening.
I'd give them my card.
Well, what an honor and a privilege to have moderated this, but I have not forgotten the homework. So hopefully you all have written your letters, the imaginary letter. Let me come to all of you to hear what you have written to your families that is going to be opened in 25 years. Mia, do you want to start?
Yes, that we can prevent Alzheimer's disease, and brain health is the priority throughout the whole life.
Fantastic. Rhoda?
That our focus now is on precision brain health and optimizing people's brain health across the lifespan so that we don't have to use precision medicine.
Miranda?
I would say, believe it or not, but in 2025, Alzheimer's disease and dementia was the most feared health condition of growing older, and it no longer is.
Awesome.
Good one.
Howard?
Yeah, I would tell them I was very privileged and proud to spend a lifetime as a geriatrician and a neuroscientist, and I worked with hundreds of scientists to get to a point where I hope 25 years from now, Alzheimer's disease is conquered. And I believe that one metric about prevention is that if we could delay the onset of clinical symptoms and the clinical appearance of Alzheimer's by 5 years, we would reduce the number of cases by over 50%. And I do think that's an achievable goal even today. And I would hope that my great-granddaughter, or whoever it is, would appreciate the fact that the work we do today ultimately will give her a world where there is no Alzheimer's, or that the impact of Alzheimer's on people is greatly reduced.
Marty.
Well, my motivation has been precedent of something like a statin. And so I see something in the book and the letter to my children that I was privileged to be able to work on a series of things that were precision medicine oriented, novel therapies, that with all these diagnostics and biomarkers, there were options to treat the individual. And I agree, at that point, age will just be a number.
That's good.
Jerry.
I think in my letter, I would say that, throughout the majority of my life, that there was no treatment for Alzheimer's disease, and that I was thankful at a relatively young stage of my career that I got bit by the research bug, and that instead of becoming a neurosurgeon, I, started my research career, and am proud to be part of a team that's helped contributing to the cure that we're going to have in 25 years for Alzheimer's disease.
Fantastic. Frank.
So I would say that my team and I were privileged to go out into the world and look at the vast discoveries in neuroscience and harness the very best of those neuroscience and biologic discoveries to bring them to bear on the Alzheimer's issue and the brain degeneration issue, so that we have ways of preventing that degeneration.
Wonderful. Robbie.
I would say that I'm very grateful that, 20 years ago, or I guess in this case it would be 45 years ago, that there was an ADDF, and that when I came to them with the idea that we could regenerate the Alzheimer's brain, they said, let's do it. And I'm very grateful and proud to be a Goods Prize winner, and to be a member of this esteemed group of innovators that keep on innovating.
Jeff.
So my family would say Jeff understood that science is a relay race, and he was able to pass the baton of passion to the next runner.
All right. Well, congratulations to all of you. Your dedication and commitment is what makes us confident, optimistic, gives us a lot of hope that we're going to solve this problem. Please join me in giving them a warm round of applause. Well, thank you for indulging me. Thank you to the Louder family. Thank you to the co-chairs, Heidi and Tom, and thank you, Nancy.
Big round of applause for Bose. Incredible Goods Prize winners. At this point in the proceedings, we're about to announce the 2025 Goods Prize winner. As a sign of welcoming from our previous Goods Prize winners, would you all don your medals right now? The medals they are putting on were actually developed by somebody from the US Mint, and it's based on the Nobel replicas, so kind of cool, right? And yeah, this is contest, how quick can we put them on? Don't knock your microphone off. Oh, this is good. So second, I want to ask ADDF's co-founder and Chief Science Officer, Howard Fillett, to come over here to the podium. He has the honor of presenting the 11th Annual Goods Prize. Howard?
Thank you, Mark. It's really an honor to be before you here today to give the 11th Annual Goods Prize Award. Changing the course of Alzheimer's disease is not something any one person can do alone. It requires collaboration and vision and time and passion, and more importantly, a dedication to the work. And I think each year that we identify a scientist for the Melvin Goods Prize, it's in recognition of their pioneering research that has changed the thinking of an entire field, has changed the way we practice in the clinic, has changed the way we do clinical trials, and how we diagnose the disease, sparking new innovations and ideas that will revolutionize Alzheimer's research, treatment, and prevention. This year's recipient, Dr. Dan Skovronsky, embodies that ethic. We've known Dan for almost the beginning. It started, one of the first things that happened at the foundation when I had a desk and a chair was getting a phone call from Dan's group at the University of Pennsylvania talking about an idea that they had for a new brain scan that Dan happened to be on the team and was developing. The impact of this brain scan cannot be underestimated. It was termed by a leading neurology journal, a journal called Neurology, as a revolutionary progress. And that was really Dan's idea back in the early mid-90s. Last summer, Dan and his team at Lilly received approval for their drug Kisunla and began making it available to patients in a historic moment for the world and for Alzheimer's disease. In large part, the development and marketing and availability of these new drugs for Alzheimer's disease, for the first time in human history, we have drugs that can change the course of Alzheimer's disease, and they're reasonably safe, and they're going to just get better. But that advent of new drugs was why we were so interested in biomarkers. It was one of our pillars from the very beginning. But it's particularly important why the biomarker that Dan developed, the PET amyloid scan, was critical, not just to early diagnosis, but why we invested in it. It was to accelerate drug development. And the first FDA approval of any drug that was disease-modifying for Alzheimer's disease was based on the data that showed that you could remove the amyloid completely from the brain, and in association with that, that people had a slowing of the rate of their progression. This was just amazing to see that a drug could remove the plaques from the brain, to be able to see that in living people without having to do brain biopsies or even autopsies, which most of my patients didn't want to get. So Dan has really changed the world in his group. And he's been at it, again, we met 25 years ago, and we followed his historic meteoric success, and we're very proud of him and really glad that Dan could be part of our community. It takes a long time to find success. It's said that 90% of the people that work in the pharmaceutical industry spend their entire careers never working on a drug that comes to market. But what enables that, in large part, again, are the kind of biomarkers that Dan developed and brought to market. Dan is also interesting because he not only worked on the science when he was at Penn, but he decided to spin out a biotech company and became a biotech entrepreneur. And that program, that biotech, was then bought by Lilly, very successful and entrepreneur, joined Lilly, and now has skyrocketed to the top leadership at Lilly. So he's really the icon, I think, of what a successful career can look like in both being in academia, being in the biotech industry, being in the pharmaceutical industry. So I can't overstate really how extraordinary Dan's achievements have been and the role that he's brought to our field in not only the biomarkers, as I've said, not only in accelerating treatments, but also in prevention. There are prevention studies going on now where Lilly, for example, is going out into the community and doing blood tests and then scans, identifying people who are normal and have not even developed the positive brain scans yet, and seeing if by giving these people drugs, we can prevent the development of plaques and tangles. And presumably that will delay the onset of Alzheimer's disease, which is something we want to do. Let me just close by saying that I'm really honored to be awarding Dan, a colleague for many years, this prize. And it's in name of, let me remind you, Mel Goods, who is really an icon in the pharmaceutical industry for what he did. He served as CEO of Warner Lambert back in the 90s and brought the game-changing drug Lipitor to market. But before he brought Lipitor to market in 1993, he came across a drug called Tacrin. Tacrine is very similar in mechanism to drugs that have been on the market since about 1995, which are cholinesterase inhibitors that are symptomatic agents. But Mel recognized the coming epidemic of Alzheimer's disease, and he actually brought the first drug for Alzheimer's disease, a drug called Tacrine, to market because he wanted to show that there was a way. And he did that because most of the drugs that followed him, up until, as Jeff mentioned, we had many, many failures for many years. But the drugs that were on the market all those years were cholinesterase inhibitors like Tacrine, which Mel was the first to bring to market. I had the privilege of knowing Mel personally and met him many times with the family and with Nancy. I worked with them as their physician. We went to Scotland to get some drugs to try to help Mel slow down the disease, and worked with them on caring for Mel and his long journey, his very long journey that Nancy was very committed to, caring for Mel in a truly excellent way. And so Nancy and the family established this award to create a meaning from the struggle that Mel had and his knowledge of drug development to support the brightest ideas in the hope that one day, even if it would be too late for Mel, someone would bring a drug forward that not only could treat this disease, but prevent it. And I think that's going to be possible in the coming decades. So Mel lost his fight with Alzheimer's disease last year after suffering 15 years with the disease and being cared for by Nancy. And I can tell you 15 years is a long time, even for Alzheimer's disease, to suffer the gradual and progressive, inevitable loss of function that makes us all human. Mel suffered a long time to that. But tonight we're fulfilling Mel's dream by presenting the annual Goods Prize Award to Dr. Daniel M. Skovronsky, whose drug offers hope to millions of people around the world. Please join me in welcoming Dan to the stage and congratulating him on this amazing achievements throughout his career. Dan, this is really a pleasure. Congratulations. And you really deserve this. I don't want to knock your thing off there, so be careful. And it's all yours. That's yours. You okay?
Yeah, thank you.
Okay.
Thank you, Howard, for those kind words. And ladies and gentlemen, esteemed prior award winners, distinguished members of the scientific community, the Lauder family, and all of us here tonight, the friends and family of the Alzheimer's Drug Discovery Foundation, thank you. It's a profound honor to accept the prestigious 2025 Melvin R. Goods Prize, an honor made even more special by the presence of Nancy Good, who shared with me loving stories of Mel's amazing life and accomplishments. I feel deeply grateful to the many researchers across the Alzheimer's disease community, including my own teams at Avid and Eli Lilly and Company, some of whom are here tonight, who we've worked together over many, many years. Our hard work and dedication to improving the lives of patients all over the world is what continues to fuel me every day when I wake up, walk into Lilly, and engage in the fight against this terrible disease. And while the 2025 award may be given in my name, it recognizes our community's entire body of work because it's impossible for anyone to beat this disease alone. I also want to thank the ADDF, not just for this award, but for your unwavering dedication to combating Alzheimer's disease. I wouldn't be here tonight without Howard's and the ADDF's support of me and some of my wildest ideas more than 2 decades ago. When I was a young scientist working at the University of Pennsylvania, trying to develop new ways to diagnose Alzheimer's disease, ADDF made the decision to fund my research. Although at that time, we hadn't yet seen the potential of this technology in human trials. In fact, we hadn't even gotten it to work in animals yet. Howard and the ADDF were willing to take a risk on an unproven young scientist that they believed someday could change the world. Because of that grant, I was able to pursue research at the university that eventually led to the creation of a company built on a shared vision that through early detection and early intervention, we could someday prevent Alzheimer's disease. That company went on to hire more than 100 scientists and invested nearly $100 million to eventually find success in creating the world's first FDA-approved amyloid PET tracer. That amyloid imaging agent, now called Amyvid, transformed how we conduct clinical trials in Alzheimer's disease and made it possible to run successful trials of amyloid targeting therapies at Eli Lilly, where our forces swelled to thousands of scientists and billions of dollars invested against Alzheimer's disease. Because of the ADDF vision, more than 2 decades ago, and on the back of that first initial grant, we now, for the first time, have approved therapeutics that can clear amyloid plaques in Alzheimer's disease and slow the course of decline. That initial grant from the ADDF not only opened up the field for therapeutic development, but it also catalyzed the development of next-generation Alzheimer's diagnostics. Because of our success with Amyvid, we were able to create TauVid, the world's first tau imaging agent. And then we went on to create the phospho-tau 217 blood test, which at Lilly we're now using to enroll people in trials who don't have any symptoms of Alzheimer's disease. But we know from the blood test they already have the first signs of the pathology in their brain. We're treating them with experimental therapies, hopefully clearing their brain of amyloid pathology years before the disease has robbed them of a single memory. We're only a few years away from knowing if that trial works and we can actually prevent Alzheimer's disease. And that, ladies and gentlemen, is the power of helping a young scientist and startup entrepreneur whose company ended up getting bought by Lilly and changed the trajectory of my life and the lives of countless Alzheimer's patients forever. That is the power of the ADDF. While this award celebrates what has already been accomplished, we must remain focused on the next breakthrough, the next treatment, the next discovery that will transform patients' lives. To that end, while we've had success in recent years with disease-modifying treatments being developed and approved, we still have a lot of work to do as the diagnosis, treatment, and care paradigm for Alzheimer's patients aren't where they should be. At Lilly, we're committed to pushing the boundaries of what's possible today, while also investing in research, some of which may seem far-fetched now, that could become tomorrow's game-changing treatments. For example, we're turning the clock back even further, moving to primary prevention before the pathology of Alzheimer's disease starts. We're working to diagnose and target each of the several pathologies that we know can cause neurodegeneration, including amyloid and tau and synuclein and others. Within the next decade, I expect with new diagnostics and new therapeutics, we'll be able to stop neurodegeneration even before it starts. But that's not all. We're working at the RNA level to turn off the genes that we think cause Alzheimer's disease. And we're even working to edit the DNA that we're all born with and remove the risk of neurodegeneration even before it started. That's the promise I make to my children. And indeed, for my family, like for many of yours, the fight against Alzheimer's is a personal one. Tonight, I dedicate this prize to my grandmother, Olga. She was an amazing woman. She survived the horrors of Auschwitz. And she was so strong, no obstacles in life could stop her. Nothing could hold her down, except for Alzheimer's disease. I remember the day she died. I was in France, setting up a clinical trial for a new Alzheimer's therapeutic. And I thought, someday I hoped I would make her proud. Of course, our work was too late for her, but not for my parents' generation, certainly not for my generation. And for my children, I think Alzheimer's will not even be a concern. So to all of you, whether you're current or future scientific pioneers, entrepreneurs, academics, physicians, patients, caregivers, or generous supporters, we're all called to confront this challenge together. No single one of us will find the answers on our own, but together there's no limit to what we can accomplish on behalf of patients. Together we can and will make history and change the world yet again. Thank you once again for this incredible recognition of my work. This will forever be one of my most cherished moments. Thank you.
Please welcome back to the stage Dr. Sanjay Gupta.
All right. That was fantastic.
Thank you.
Congratulations. The Nobel Prize of Alzheimer's Medicine.
Awesome.
Congratulations, yeah. And it's nice to be with the Avengers of neuroscience here.
Yes, I see there's an empty chair there.
All right, exactly. Thank you again so much for having me, and thanks to the Louder family. You've done something really special here. I mean, I've been reading about this stuff for a long time, and I learned a lot tonight already, so thank you. I really appreciate it. Dan, you got into this a little bit, but so you're a student at Penn, and you are thinking about how to basically diagnose or at least screen for Alzheimer's disease. How did this come about for you, and why ADDF?
Yeah, sure. I actually was studying neuropathology. I'm a trained neuropathologist. You of course know what that is. We're the ones who look under the microscope after a surgeon like Dr. Gupta takes out the tissue. In the case of Alzheimer's patients, unfortunately, we do that after they've died. We look at their brain under the microscope to try and figure out what they had. Although that was my training, I didn't love it. It obviously is too late for the patient and their family. And so that got me curious about whether the same things we see under the microscope to make that diagnosis, if we could do that in living patients. And I was lucky to team up with a chemistry team that we tried to take those same kind of dyes that we use under the microscope, and turn them into imaging agents that we could inject into patients. And ultimately that was successful.
And where in life were you at this point?
I would have been a graduate student when I started this, and 20-some years ago, I wrote a PhD thesis that said someday we're going to be able to image amyloid and then test drugs that remove it, and I predicted they would work.
And maybe, Howard, you can give us some insight then. So you take a call like that, or the organization takes a call like that. It's very early. There's no sort of human validation of this yet. What makes you jump on board?
Well, I thought it was a good idea.
I mean, to be fair, though, you probably got a lot of good ideas.
Yeah. Well, the problem in due diligence, we see about 300-400 ideas a year for new drugs for Alzheimer's disease and new diagnostics. Bose mentioned the Diagnostics Accelerator, which actually has a speech program. The Lauders were really visionary in how they set this up, because the initial letter that we wrote to the IRS for incorporation included biomarkers. That we told the IRS we were going to do one thing, and that was accelerate the development of new drugs. And we were going to do that by developing new biomarkers, by doing treatments, developing new treatments, and by prevention. And that's been our North Star from the very beginning. We don't do education or advocacy on the Hill or anything else except developing new drugs. And we told the IRS that we were going to be a venture philanthropy, which at the time was almost unheard of, essentially unheard of, that we would be investing in biotech companies, because if you're going to be developing drugs, you need to partner with industry. You need to go to the small biotechs like Dan spun out, Avid Radiopharmaceuticals, based on the work we funded at Penn. And then, of course, Avid had to be taken up by Lilly to really do the kinds of very expensive large studies that would validate this as the first diagnostic test in the history of the world for Alzheimer's disease.
And how widely is this now adopted, Dan?
Oh, quite so. It's available everywhere in the United States and around the world. And now it's routinely used, I would say, for most patients before they initiate therapy.
And who is it always people who are early symptomatic, or is it even for people who have strong family history or genetic predisposition? Who should get this test?
Yeah, so the way that it's used today in medical practice is people who are symptomatic and the doctor's working them up to find out if they have Alzheimer's disease or not, usually in the context of making a decision to start therapy. Of course, in research settings, we use it more broadly than that, including to identify patients, as I was saying before the symptoms start. Now, it's a little cumbersome to get people like any of us in this room who don't have any symptoms, but maybe we're worried or curious about our risk for Alzheimer's to go do a PET scan.
Yeah.
So we use that PET scan to validate and create a blood diagnostic that can give us almost the same information.
So yeah, I'm curious, Howard, who should get the blood test then? I mean, we're talking about this testing, and we've had quite a long discussion about it tonight, but what does it mean for the average person?
Well, there's two kinds of people out there. Of course, it's a continuum, but there are people that want to know. I mean, I've seen 50-year-olds and 60-year-olds where they've seen their loved ones suffer from Alzheimer's.
So people who have no symptoms necessarily, but they've seen it.
But they want to know their risk.
Which is the situation with Dan, with your grandmother, and my grandfather.
And you know, I say to them, well, you know, we can do your ApoE testing to see if you're EE4, which means you're going to get it.
So that's the first thing you recommend?
Well, it's one of the genetic risk factors for the disease, and people tend to get it 10 years earlier. So instead of 75 people with EE4, get it at 65. So that's one thing that I would do in terms of doing tests to look at their profile in a patient who's worried but not symptomatic. And then there are people who don't want to know. They say, you know, if I got that test and it was positive, I'd just be so anxious for the next 10, 15 years that it's just not worth it. I don't want to know until I get symptoms. It's a different equation. So I think it's really, we're going to have guidelines from the experts and the leaders around the world about how to use these tests. They're already somewhat out there. But I know in my practice, where I'm practicing medicine, and that's why we call it practice, we're dealing with individual people. We're dealing with patients where I have to describe to them the risks and the benefits of getting the test and what it's going to mean. And in a world where we have drugs, they'll have the option, at least, of deciding on whether or not to take therapy.
It's interesting because you used the sort of metaphor of a cholesterol test earlier, you used the metaphor of a blood pressure cuff. I mean, these are screening tests. I think most people in this room have gotten their cholesterol checked. Do you think we're going to get to that point?
I don't know if we'll be screening, but you know, the PET scan costs about $8,000, and Medicare has recently decided to pay for it, which it was approved by the FDA in 2012, I think it was, or 2014. I think I got that right. Around that time.
It's funny, nobody could remember.
Yeah.
Memory jokes.
Yeah, right.
Yeah.
But it hasn't been widely utilized because it wasn't paid for by Medicare. I mean, here's a diagnostic test in that context that has high degree of sensitivity and specificity. It's a good test, and Medicare wouldn't pay for it because there's been bias against Alzheimer's disease, essentially. And I think part of it was Medicare was afraid that it would open the floodgates of testing and so on. Whereas the blood tests are going to come in around $500 or $1,000 or maybe even $1,500 and give us the same information pretty much. You don't get the regional distribution that you see on a PET scan, but that's not as important really as getting a positive or a negative answer.
I will tell you, I worked on a documentary on this, and as part of that, I ended up getting my markers checked, and it's a really interesting sort of meta experience to actually do it. I don't quite know what to make of all the numbers yet, but still having this insight into something that's happening in your brain via a blood test is really interesting. Dan, I'm curious, with all that you know about Alzheimer's, but also around GLP drugs, which came up, and I should point out that I interviewed Dan for another documentary that I worked on. He was a star of a GLP documentary that we did. What is going on here? Why is it that GLP drugs might have such an impact on Alzheimer's disease?
Yeah, it's just super interesting science, and at Lilly we've developed GLP-1s, including a dual-acting incretin called tirzepatide that's marketed as Mounjaro and Zepbound, and we're really interested in all of the health benefits that might happen to patients as they lose weight. We know that diet, what you eat, is so central to your health, and as a medicine like this changes your diet and what you eat, it has a myriad of other effects. As was commented earlier, we see inflammation in the body dropping. We see the risk of other diseases. The risk of developing type 2 diabetes goes down. Sleep apnea clears up. Liver disease goes away. All of these benefits are related to the primary mechanism of action, I think, which is changing your metabolism from an unhealthy, fat-accumulating, inflammatory state to a more healthy, fat-burning or lean or metabolic state. How does that affect the brain? I think we're just at the beginning of finding out. As was mentioned, there's a trial ongoing with one of the other incretins, semaglutide, in Alzheimer's disease patients. I think one of the things we believe is that these kinds of medicines, by improving your cardiovascular health, will decrease small strokes that are very common. And we know that when people die, if they have Alzheimer's pathology and small strokes, they had a much more rapid progression. So maybe if we can improve their vascular health and also remove their amyloid plaques, that could be the best outcome. Those are the kinds of experiments we'd like to do in the future.
You know, Howard, I'm just curious, we have just a little bit of time. With all that you've learned over the last quarter century, and based on some of what Mia talked about as well with regard to the data now on prevention, how do you live your life differently?
You know, I think even for myself, I try to be compliant with a healthy lifestyle. But, you know, we work very hard, and we're often on the road. And it's difficult for people, I think. That's another reason for getting this preclinical diagnosis. If I knew I had plaques in my brain, or if I was an ApoE4 double homozygote, it might give me, if I wanted to know, and that was a positive thing, it might give me more motivation to be compliant with the finger type of approach, with exercise, which I've been doing all my life, and diet and social activity and cognitive training and managing diabetes and hypertension. So I think there's also that side of knowing also. You know, people can try to do what they can do. It doesn't mean, you know, you get sometimes a narrative from a person that there was a famous person and he lived a healthy lifestyle and he got Alzheimer's disease anyway. And we don't know for that person whether they might have gotten it 10 years earlier had they not been compliant with a healthy lifestyle.
Sure.
So I think it gives incentive. On the semaglutide story, which I think is really interesting, I just had a couple of comments if I could. Well, one is that although the drug seems to affect multiple organs, all the organs need effective glucose utilization. And as we age, insulin resistance becomes a common effect in most people throughout the body. The thing about the brain is that it's only 2% of the body weight. It uses about 20% of available energy through glucose at any given moment. And the most metabolically active part of the brain is the hippocampus, which is where Alzheimer's disease starts. So imagine you're eating a regular diet, but the glucose in your diet is not getting where it's supposed to be. And we know that in diabetics, for example, if they take too much insulin, what's the first thing that happens is they go unconscious. That's how dependent the brain is on glucose as a form of energy as well as oxygen. And we recognized this probably 10 or more years ago and funded a study of liraglutide, which was a precursor to semaglutide. And just to give you an example how partnerships can evolve and work, we decided to fund a study of liraglutide at Imperial College of London, an investigator named Paul Edison, in people with mild cognitive impairment. And it was a small study. I think we gave them like $1 or $2 million at the time. Well, when Novo Nordisk heard about this, liraglutide was also their drug, so they wanted it to be a really good study. So they came in with an additional $7 million or so to partner with us, get a seat at the table, help design the study, and look at the data. And then General Electric came in and they said, well, you guys are going to need the amyloid scan, and we're willing to donate the scan, which Lilly does a lot for studies, for research with the Amyvid scan. And so we'll donate the amyloid scan through their product, which is related to the Amyvid product that Dan invented. And so it became a much larger study. It was a focus of collaboration. The results were not definitive, but they were interesting enough that Novo Nordisk decided, we got this new drug, semaglutide, and we're going to spend something like $500 million to see if it works in Alzheimer's disease. And so it was that kind of seeding the innovation from a small, relatively small foundation, I mean, relative to what Big Pharma can do. But we have to work together to move these things forward.
I love the story. I think the story of how these scientific achievements are created are some of the greatest stories that we get to tell. So we're out of time, unfortunately. But thank you very much, and how about a warm round of applause for our panelists?